In this screencast, I'm going to go through the cellular respiration virtual lab. If anyone is having issues, they can refer back to this as I work through it. I have received emails that a couple of students are struggling with their computer accessing it. So I have right here the link to the actual write-up. So you are going to want to annotate it in Cami. So you're going to want to read through the background, carbon dioxide oxygen balance, and now we're going to do the carbon transfer through snails and Elodia. Now, you're going to have to go to this, I linked it as well, I believe, but you're going to need to go to the website that's listed. It might tell you to upgrade. I'm going to allow it. We want to allow Flash Player. In December, this is going to be unavailable, but for now, we're okay. And you're going to do carbon transfer through snails.
You are setting up a snail aquarium at home. Your aquarium kit contains a bag of snail eggs on an Elodia plant. The instructions say to pour the contents, including the Elodia, into the aquarium. But you're not sure why you need the Elodia. Why might Elodia plants be important in maintaining a healthy ecosystem? In this investigation, you will use snails and Elodia to explore a biological system. You will form a hypothesis about the relationship between snails and Elodia, and then design an experiment to test your hypothesis.
Okay, so that's our problem.
Explore the lab to learn what is available for your investigation. You must click on each item in the checklist. Once you are ready to move on, click Procedure.
Okay, before I start doing that, I did want to show you that if you accessed it here, sometimes it asks for what state you're in. So make sure that you just say Indiana and it should let you proceed. Okay, so read through the problem tab, create your own question for what you are investigating in this experiment. So the problem, you're just going to restate the problem in a question form. And then you're going to explore the lab to learn what's available to you in your investigation. You must click on each item in the checklist. Okay? So we're going to go back here. The beaker of bromothymol blue, BTB solution. And if you click on it, I believe it will tell you.
Beaker of bromothymol blue, BTB solution. Bromothymol blue, BTB, is a chemical indicator used to test for the presence of carbon dioxide. This BTB is green because there is already some carbon dioxide in the solution.
Okay, so I'm going to go ahead and close that out. Rewind and watch this if you needed to see what the bromothymol blue is and why it's green, but you should be able now to answer part A. Okay. So I'm just going to go through and click on all of these, and then you can go back and forth with the video. So the pond snails are in the speaker here.
Pond snails. Pond snails have lungs. They can breathe in or out of the water.
Okay. Now this is the Elodia.
Elodia. Elodia is an aquatic plant.
We need test tubes.
Test tubes. Use the test tubes to hold the snails and Elodia.
Next is the grow light.
Grow light. Use the grow light to simulate light conditions. If you choose to leave your experiment in light conditions, you must expose the organisms to a constant light source.
Test tube rack cover.
Test tube rack cover. Use the test tube rack cover to simulate dark conditions. Carbon dioxide oxygen cycle poster. Use this diagram to help you write your hypothesis. Assume the cycle is true for snails and Elodia.
Okay, so what it means is that this is with a mouse and a plant, but assume this is it applies as well for a snail and the Elodia, the plant. And the
color key. Use the key to determine the level of carbon dioxide in a solution.
Okay, So we have now clicked on everything here in the checklist. If you need to stop and go back, pause the video so you can answer A through E. For F, you're going to draw the picture. Just a quick diagram is fine. And then fill in G based on the color of the, what do they call it? The color key here. Okay. Now, this has you do an if-then statement. My thoughts on if-then. You don't need to. If you would rather just put a hypothesis in a statement form like we've been learning about it, that is fine. That is completely fine. You can just type it out. You can just type it out anywhere on it. Okay? And then answer the questions about independent and dependent because you're going to conduct an experiment. Okay? You will be using 8 test tubes for this experiment. Fill your test tubes and perform the experiment according to the data chart found below. So it's going to have you set up the first rack just like this. Okay? So test tube 1 is your control, 2, 3, and 4, no snails, no Elodia, 2 snails, 2 Elodia, and 2 snails, 2 Elodia. So we're going to have to set that up. So now I'm ready to go on to my procedure.
Open the lab notebook and write a hypothesis.
You don't need to. You're writing it on
how carbon dioxide cycles in aquarium water
PDF.
Through snails and Elodia. Review the background information or the poster if you need help with your hypothesis. Once you have written your hypothesis, click the arrow to advance to step 2.
Okay. So just really quickly, here's the background.
Organisms are dependent on a healthy carbon dioxide-oxygen balance. Photosynthesis and cellular respiration are key processes in maintaining this balance. Plants, through the process of photosynthesis, use energy absorbed from sunlight, water, and carbon dioxide to produce carbohydrates and oxygen. Animals and plants, through the process of cellular respiration, use oxygen and sugars to produce carbon dioxide, water, and the energy needed to maintain life.
Okay. So here's the lab notebook, but like I said, you should be able to just do it in the PDF. If it doesn't allow you to do that, just type anything in here, okay? I don't want to print. I just want to close it out. Okay. So now I can go to step 2.
Open the lab notebook and design an experiment to test your hypothesis. You will begin your experimental design by establishing your variables and control. Once you have answered the questions in the lab notebook, click the arrow to advance to step 3.
Okay, so you might have to type in here just to advance it, and that's fine. Okay.
Begin your experiment by dragging the test tubes to the test tube racks. Drag the beaker of BTB to each test tube. Once you have filled each tube, click the arrow to advance to step 4.
Okay, 1, 2, 3. Oops. Come on. 4. 1, 2, 3, 4. Now I have to fill it. 1, 2, 3. Okay. Now step 4.
Add snails and Elodia to the tubes according to your experimental design.
And remember, your experimental design,
we're
actually setting it up in the PDF.
Start again. Click Clear Tubes. When your setup is complete, open the lab notebook and record your predictions.
Okay.
Once you have completed your prediction for each tube, click the arrow to advance to step 5.
Okay, so the first test tube rack right here, we're going to do nothing, 2 snails, 2 Elodia, and 2 of each. Okay, so we'll leave number 1 is nothing. So 2 snails, 1. 2 Elodia, and 2 of each. Okay. And then you're going to write down what color. This one's going to be grown under the light, and you're going to predict what color it is. Okay. Remember, the color is based on the BTB solution, and the color key, it determines the amount of, or it indicates the amount of carbon dioxide. So the second one, it looks like we have it set up the same way. So we'll leave nothing, or put nothing in that one. Not 2, 3. 1, 2, 1, 2. And so make sure that you fill out your color predictions before, so pause the video before you go on, okay? I'm then going to, oops, I don't think I can do that yet. Don't think I can do that part yet, moving the box. Okay, and so this is, yep, and this is your setup here. Go ahead and type out, you might have to, again, just put anything in here so it will forward it for you. Okay. Let's go on to step 4.
Double check your setup, then click Add Stoppers to stopper the tubes. If you are testing in light conditions, drag the rack to the grow light. If you are testing in dark conditions, drag the rack to the cover. Click Start to leave the tubes for 24 hours. Once 24 hours have passed, click the arrow to advance to step 6.
Okay, so we're going to add stoppers. I double check. They look good. We'll add stoppers here. Now, remember, the first one is going under the grow light, and the second one is under the test tube cover. So we'll take these and put it under the grow light. We'll take these and put it here. Drag the rack to the cover. This might be the issue that people are referring to. Let me try this one. There we go. I guess you just have to get it just right. Okay. So now we're going to hit start and go for 24 hours. Okay. Now we're going to click the arrow to advance to step 6 here.
Observe the change in color of the BTB solution in each test tube. Open the lab notebook and record the results of your experiment.
Okay, so
Once you have recorded your results, click the arrow to advance to step 7.
Okay, so you're going to look at each test tube here. This is the one in the dark. This is the one in the light. And you're going to write the end color down, okay? Whether it's yellow, green, or blue. And sometimes you just have to use your best judgment. Okay. Now.
You are now ready to analyze your data and draw your conclusions. Open the lab notebook and answer the questions.
Okay. And so this last part here should be similar to the questions found here. So at this point, you do not need to add or do anything else with your lab notebook. We're on the last step. And you guys are ready to make some conclusions about it, okay? So you'll need to use this and the colors here, and you're going to try to interpret why they are the colors they are and see if it makes sense to you. So hopefully, if you cannot access or if you're struggling with your computer using this program, this screencast can help you complete your assignment.